Hi, my name is Anna Kelley, and I'm joined here today with my two peer reviewers, Ella Milligan and Ellie Leandris. I am presenting um, my project on a high energy, low cost plasma device for chemical degradation. So before I begin talking about my procedure, I just wanted to go over the two main ideas, which is plasma. And basically plasma is just the fourth state of matter. It makes up most of our universe. It has a diverse amount of applications, such as plasma screens, CFL lighting, microelectronic developing, and what I will be focusing on is removal of pollutants. One problem with plasma is that it requires a lot of voltage. And so if you wanted to use plasma somewhere where you can't take high voltage, that's a problem. But one solution for this is piezoelectric crystals. And that brings me to my research question, which is, will the polarity of my hand cranked plasma device trigger different chemical modifications? So first, I'm going to talk also about how piezoelectric crystals work. Piezoelectric crystals have the ability to transform mechanical energy into electric energy, as well as vice versa. So a perfect example of this would be a light up sneaker. If you've thought about, the kid's shoes, you step on it, and that gives energy to the light bulbs in the shoe, therefore lighting them up. So here is my setup. And so my hypothesis is that I'm claiming that the polarity of plasma can trigger different chemical degradation levels on the surface. So what we're looking at here is the difference between the positive electrode and the negative electrode and how that affects dye on a piece of paper. So here is just a close-up of the device tungsten needles. Here on the left, you can see the tungsten needles with the plasma off. And down on the right is the plasma on, which is me cranking the device. So my experiment procedure was simply taking regular white printer paper and marking the distance between the two needles. I put a drop of brilliant blue solution mixed with some water. I would then put a drop of that on the 2 points and place that under the needles. And I would crank for 5 minutes with applied plasma. I would take a picture and I would crank again, take a picture and crank again each 5-minute periods. So here is the pictures I took after each 5 minutes of applied plasma. On the top left, you can see before any of applied plasma. Right under that, 5 minutes of applied plasma. The top right is 10 minutes of total applied plasma. And the bottom right is 15 minutes total of applied plasma. Each 5 minutes was about 600 cranks, which I counted with my phone while cranking. So this is just a close-up comparing before any applied plasma and then the 15 minutes total applied plasma. From the results, we can see that while both polarities had an effect on the blue chemicals on the surface of the paper, the right electrode's color change is more evident. In the future, to improve the efficiency of this device, ideally, I would be able to construct a prism-shaped crystal that has a low length to width ratio, as well as a low mass to cube length ratio. This is based on literature reviews that I've done. And also because my conclusions were subjective and I simply looked at the left and right electrode to see the color change. I'm going further and using MATLAB computer programming to analyze the color and see if I can get more of a quantitative answer. Do you guys have any questions?
Yeah, I actually have a question. That's really interesting, especially how you're going to do that with MATLAB. I think one thing I had a question about was how exactly does plasma break down different pollutants? Just like, especially with the chemical structure of a pollutant, how exactly does the plasma help break that down?
So the chemical reaction we're looking at here is simply just oxidizing. It's taking the air and it's high voltage, and so that's breaking down the chemical structure of the dye and oxidizing it. Any
Okay,
other questions?
thank you.
Yes, yes, I have a question as to how come the 5-minute interval did not look very much different from the 15-minute interval?
Right. So this experiment was put together so that I would be able to do it at my home. Due to COVID, I wasn't able to get to labs. And so basically, ideally, if I had been in a lab or really testing the efficiency of this device, I would use it on a liquid form, because as you can see from the pictures, the plasma doesn't cover a lot of surface area. And so it's only gonna degrade so much color in one little space. So if this were to be used on a liquid that's constantly moving, and yeah, the surface area is changing.
Thank you.